Hello, my name's Eli Jones, and in this video I'm going to be talking about my second phase towards the total synthesis of Lindbergin E. To start, we need to understand what a total synthesis is. This is essentially a way to convert a very simple molecule, such as a component of crude oil, into something that's synthetically useful, a very complex drug or molecule. I'm going to be talking more about this process of converting something so simple into something so complex in the future. But first of all, we need to understand why it's actually useful. To start, total synthesis allows us to make more of a given drug. Whenever a drug is discovered for the first time in nature, it's often present in very small quantities, and we need total synthesis to make more of it. Also, total synthesis is basically like molecular construction, and whenever we explore new routes to a molecule, we also discover these new construction methods, and these can be applied to future molecules. So, why did I decide to synthesize Lindbergin E? Here's the structure of Lindbergin E, and the first thing we should understand is that Lindbergin E gives us a perfect opportunity to explore one of those new molecular construction techniques. It has something called the PDAP moiety, and this moiety right here has never been synthesized to date, and is present in over 15 different natural products, just a few of which are shown here. So essentially, if I'm able to find a good way to make this structure that's highlighted in orange, I can then incorporate that into the synthesis of a variety of other natural products that are also medicinally relevant. Lindbergin E also has a very interesting medicinal property called leishmanicidal activity. Let's talk about what leishmaniasis is. Leishmaniasis is a disease caused by the bite of a sandfly infected with the deadly protozoa Leishmania. These protozoa result in the formation of very painful ulcers and sometimes internal organ damage. This can often be fatal, with over 20,000 deaths being observed each year, over 12 million cases, and over 1 million new cases each year. So the rates of leishmaniasis are growing. Right here you can see the leishmaniasis global distribution, and you'll notice that it primarily affects the subtropics where sandflies are prevalent. Also, leishmaniasis predominantly affects third world countries. So this leads us to our next point, that the current treatment options for leishmaniasis are not effective for an area so socioeconomically deprived. They're too expensive. Right here is amphotericin B, the most common treatment option, and it costs over $50 for a single injection. That's simply not feasible for these countries with such high infection rates. So, Lindbergin E might just present an alternative. With a median lethal concentration of 13.6 micromolar, Lindbergin E is the perfect candidate for exploring new potential drugs. So, we previously explained why a total synthesis of Lindbergin E is needed, but what does this actually look like? Well, a good analogy for this is building a house. You start with your foundation, you gradually build up your walls, and then you combine the roof and the main structure, and then you add the windows and the doors, and then you finally paint your house, the fancy details. Now, synthesizing a molecule is very similar. In this case, here are the two precursors to my total synthesis of Lindbergin E. And I'm actually performing what's known as a convergent total synthesis. So just like when we build a house, we make the roof and the foundation and walls separately, that's what I'm doing. I'm just doing the same thing with a molecule. I'm making my molecular roof and my molecular walls and foundations separately. The molecule on the left is a fragment A precursor, while the molecule on the right is a fragment B precursor. So you can see here, this is very similar to building a house. I'm just adding little structural details like carbon rings and carbon chains along the way, and then I finally bring everything together, just like I bring the roof and the main structure together. And then lastly, I add the fancy details, kind of like the paint. I just remove a few protecting groups. And there we go, we have Lindbergin E. Alright, let's take a more thorough look at this total synthesis. So in phase 1, which was last year's research, I focused on synthesizing fragment A, which you can see on the right. I first performed a 2H chromine synthesis using a phenylboronic and benzoic acid co-catalyst system, and then I acylated that product to obtain my final fragment A. In this year, in phase 2, I've been focusing primarily on the fragment B precursor. These are all the steps I performed so far. I started from fluoroglucanol, and I formulated that using a Vilsmeier hack formulation. I then reduced that using a Clemmensen reduction. Then I added an acyl group using a Friedel-Crafts acylation, and finally added another formal group to yield my fragment B precursor. So in phase 3, which is future, I'm going to be combining these fragments A and B together to get my final Lindbergin E. I'll start by treating the fragment B precursor with a base and prenyl bromide, and that's going to be my asymmetric alkylation, and then I'm going to combine that fragment B with fragment A in a 3-component reductive alkylation, and then finally I'm going to deprotect that resulting product using boron tribromide to obtain Lindbergin E. Okay, so we previously talked about some of the actual synthetic planning that went behind proposing the steps. However, let's talk about the techniques and equipment used. So whenever I performed this first step here, the Vilsmeier-Hack formulation, I didn't actually characterize the yield. I just went straight to this product, and then I successively performed a Clemmensen reduction. Now the overall yield of these two steps was 23%, which is pretty good. And then I performed a Friedel-Crafts acylation of that product with 54% yield. And finally, I did a Vilsmeier-Hack formulation with a 37% yield. In the lab, after running a reaction, I need to be able to actually characterize the exact product I've obtained. This is where purification comes in hand. You can see in the video I'm preparing a column, which is basically a really fine layer of sand that helps me separate the crude components from my reaction mixture. This gives me a very, very pure product, up to 99% purity, and allows me to characterize my products in NMR and figure out their exact structure. This is a very key aspect of my research.
One of the most important aspects of research is confirming the structure of my product. In order to do this, I use NMR, or nuclear magnetic resonance spectroscopy. Essentially, NMR treats my compound with a very high magnetic field and then pulses it with a special radio frequency wave. By analyzing the decay signals of my compound, I can determine the exact product using a variety of different NMR techniques.
So to conclude, Lindbergin E has some really interesting properties, from its medicinal activity against the painful disease leishmaniasis to its interesting structural properties, which allow us to access new molecules in the future. I hope you can appreciate the work that went behind this total synthesis. And if you have any questions, please feel free to email me, and thank you.